Welcome to video 3 in our course on ST9. We're here, this third video, we'll be talking about dimension reduction, feature selection, and feature extraction. So let's first talk about what feature selection is. And that is the process by which we go from our initial set of P molecules down to K molecules, where K is much less than P. Here we're talking about molecules, but we could be talking about anything that is in particular is of interest. I'm trying to reduce that number of dimensions P down to number of dimensions K. So what we should consider is that K, those changes may reflect multiple transformations on P in linear space, depending on the approaches that we are taking, and approaches may differ extensively on that. Linear space would apply to PSEA and MDS, where these are very good at, very easy for people to interpret because they're placing distinguishable features onto a 2D plane that is easy for interpretation. MDS can cope with uh, uh, some nonlinear effects better. TSNE and UMAP tend to use nonlinear space, which is more challenging for people to interpret, especially when you're putting many dimensions on a single 2D plane. So we can kind of distinguish that PCI and MDS are better in that way in the sense that they're more easy to interpret. TSNE and UMAP should be interpreted with great caution. And here we're talking about where we have some large matrix X, where we have n samples that might be across the rows and p features across the columns. And we want to reduce that number of features to a manageable number that can tell us something about this data. So what we're trying to do is take that matrix X, so just some big table like this, where you have a bunch of samples and a bunch of features, and simplify them. And we simplify them as two matrices. So we have an n by k matrix, so it's n samples by k features. So it's a smaller subset of uh, of the one shown there, and k, which is going to be a length k by p. So by multiplying these two matrices, we can obtain x at the end uh, with some sort of error. So the W matrix is our loadings or weights for the samples n by p and n by k, I should say, and HKP is the transform data for the features k and p. And then we have our error term, and we can infer x from W and H and vice versa. Use x to obtain W and H as well. But what we consider when we're thinking about different approaches that are applied is the difference between supervised and unsupervised approaches. So supervised ones require data labels to be known in advance. Unsupervised ones do not require such metadata or structures. And what we should bear in mind is that both approaches can be used. So we might use unsupervised clustering first to identify outliers or samples that are unusual, and then supervised clustering or supervised approaches to apply a particular hypothesis to that to the labeled dataset. So they can be used in tandem. So data exploration QC unsupervised approaches are very, very good. Um, and we want to kind of obtain the total maximum amount of variation possible in our visualization or numerical summary. For supervised approaches, we want to get maximize the number of features or components that tell us about the differences between the groups. So that's the key thing. It's going to get the ones that differentiate the groups A and B, not the ones that maximize the total variation that are representative of the wider A and B data set. Um, an example, supervised models would be those using training data, and then this is tested on new data, testing data to improve performance that feeds back into parameter choice. So that iterative process, or part of testing as part of testing and training, is a kind of supervised approach because we're separating the data. There's different types of dimension reduction. It can be correlation based or projection based. For correlation, we're essentially measuring covariances to allocate the features to shared patterns. Sometimes those shared patterns are called components or dimensions. For projection based, we're trying to reflect the sample's variation, but in a new subspace using selected features that are typically merged. And here we're assuming that we're dealing with Euclidean distances between our different samples, and that there's one universal solution that is optimal. So if we have multiple solutions, we may not actually arrive at the optimal one. Um, what is advantageous about that? We can obtain information about the features that contribute to a particular component. So we can say, for if we're looking at genetic mutation data, if the 10 mutations contribute strongly to a particular effect, a particular component, we can extract them from the data, and that can tell us more about the biological mechanism behind why something might occur. This can differentiate us from other models where extracting such information may be hard. So we need to consider also what information our model can give us. A key method in this area is eigen decomposition. So the decomposition refers to the data, and the eigen is kind of the stem of a word referring to different parts of that data that we're the outputs we get from that data. So what we might do first is both feature selection first, and then then dimension reduction or feature extraction. So we might subsample the features for the ones that are filtered and valid and inform our analysis and are not just noise. And then we feature extraction. So we transform our data, our features to make new ones. So we might do some sort of normalization and or scaling to change those original features, and then proceed on to dimension reduction after that. A very common unsupervised regression-based approach is matrix factorization. So that's a very common method where we are using singular value decomposition SVD on a matrix X to produce W and H, those matrices from which we can extract more information. Typically, these are rectangular matrices. An example that we're not talking about in the course is non-negative matrix factorization, but PCA belongs to this as well. And typically, we'll talk about having K, these abstracted outputs from our results will be the K eigenvectors. So eigenvectors refer to the coordinates of our samples in each of the PCs, each of the principal components, the dimensions. And the eigenvalues refers to the importance of each eigenvector. So the bigger the eigenvalue, the more important it is. And eigenvalues reflect the fraction iteration that is explained. So we have this matrix X, we have our matrix W and H. Sometimes H is the components is referred to as the latent variables or signatures or score matrix and other similar words. And X is the covariance matrix for the PCA. And what we assume in X is that the principal components, the features within each principal component are highly correlated with each other, and the features between different components should be largely independent of each other, and they should not be correlated. What we can summarize in this area, we're talking about more practical components in the demonstration videos, is some questions you should be able to answer at the end of this. Like, how many dimension approaches are there? And maybe you can give an example of how both supervised and unsupervised methods can be used in a project. In addition, you can say more about how both feature selection and feature extraction could be used in a project. And then also what are eigenvectors and what are eigenvalues and why are they different? So that completes this section. Please see our next part on applying principal components analysis.